This is Porphyria cutanea tarda, and it is characterized by lesions which occur on the sun exposed sides, for example the dorsum of the hand. Okay, it is characterized also by painless blisters, okay, which later burst and leads to formation of crustings. In this condition, the skin becomes fragile and is subjected to repeated trauma and evidence of trauma. And it is also characterized by hyperpigmentation, which occurs on the hands as well as other parts of the body. Okay, so why does or what happens in Porphyria cutanea tarda? Okay, so this is a defect in heme synthesis. And to understand this, we need to understand what is heme synthesis. So before that, we should know what is heme. So when we hear the word heme, what immediately comes to our mind is hemoglobin, and rightly so. Okay, so hemoglobin is made up of heme and globin. Okay, so this is the structure of hemoglobin, which consists of 4 polypeptides, okay, 2 alpha chains, 2 beta chains. Okay, so it's 1, 2, 3 and 4 polypeptides, and to each polypeptide we have a heme group which is joined. So we have 4 polypeptides and 4 heme groups which make up the structure of hemoglobin. Now if you look at the structure of heme itself, now heme is made up of 4 pyrrole rings. Okay, we have 4 pyrrole rings, and these pyrrole rings have a ferrous ion at the center. So the ion binds to these 4 pyrrole rings, and the heme group also binds to the globin chain with the help of histidine amino acid. So we have a proximal histidine and we have a distal histidine, by which this heme group, this blue color heme group, is binding with the globin chain. Now when the hemoglobin is deoxygenated, so between the distal histidine and the ion we have a water molecule. Okay, but when the hemoglobin becomes oxygenated, or when the oxygen comes, this water molecule is replaced by oxygen. So this is how oxygen is carried by hemoglobin. So the oxygen is binding to the heme groups. So there are 4 heme groups. So there are 4 oxygen molecules which are carried. So each hemoglobin molecule can carry 4 oxygen molecules. Okay. Now let us see what the pyrrole ring is. So the heme group is made up of pyrrole rings. Okay, so we have, this is 1 pyrrole ring, 2 pyrrole ring, 3 pyrrole ring and 4 pyrrole rings. Okay, so the heme group is made up of 4 pyrrole rings, and these 4 pyrrole rings together they form as porphyrin. Okay, now this porphyrin is photosensitive. So the picture which we saw of the clinical condition Porphyria cutanea tarda, so that was representing photosensitivity. Okay, so why was that photosensitivity occurring? Because the porphyrin levels in the blood was accumulated, and these porphyrin levels in the blood, okay, they are photosensitive. In this, when they deposit in the skin, they are photosensitive. That leads to all those reactions and all those features that we have seen. Now these pyrrole rings, they are joined together by methinyl bridges. Okay, so this is the methinyl bridge, okay, which is joining the 1 and the 2nd pyrrole ring. So we have alpha methinyl bridge which is joining the 1st and the 2nd pyrrole ring. Similarly we have as beta, delta and gamma methinyl bridges. Okay, so when this structure is complete, we have this iron atom at the center, and then it results in the formation of heme. So porphyrin plus iron atom at the center leads to formation of heme. Okay, so heme basically is the ferrous ion at the center and 4 pyrrole rings which are joined together by methinyl bridges, as we have seen previously. Okay. Now apart from hemoglobin, the heme is also responsible for various other proteins and enzymes. Okay, for example, cytochromes. Now, cytochromes are an important component of the electron transport chain. An electron transport chain provides us ATP for energy, which is utilized by all the cells. So the cytochromes, for example, cytochrome B, C1C are important components of the ETC, and they require heme. Okay, they are made up of heme. Similarly, we have is cytochrome P450 enzyme systems, which again are made up of heme, and these enzymes are induced when we take certain drugs like antibiotics for their metabolism. Okay, we have another enzyme system like catalase, okay, which when we study antioxidants and free radicals, then we will see. So these are important antioxidants. Then proteins like myoglobin, which is a reservoir form of oxygen in the muscles, they are also made up of heme. So heme, apart from being its presence in the hemoglobin, it also has important functions. Okay. Now let us look at the metabolism of heme synthesis. Now, heme synthesis takes place partly in the mitochondria and partly in the cytoplasm. Okay, the starting material is glycine, which is a neutral amino acid, and succinyl coenzyme A, which is an intermediate of TCA cycle or the Krebs cycle. Okay, so succinyl coenzyme A, because it is an intermediate of the Krebs cycle and it is helping in the formation of heme, so this shows the anabolic role of TCA cycle. Okay, so the TCA cycle or the Krebs cycle has both anabolic as well as catabolic role. So here we are seeing the anabolic role of TCA cycle in the formation of heme synthesis by succinyl coenzyme A. Okay, so the enzyme which is required is ALA synthase. So amino levulinic acid synthase. So it is in the name of the enzyme itself, you know what the product is going to be. It is ALA synthase, so amino levulinic acid is getting formed. Okay, now this enzyme, okay, this enzyme is the rate limiting enzymes. You have two types of ALA synthase 1 and we have is ALA synthase 2. Now ALA synthase 2 is present in the bone marrow, okay, and it has a constant function. So ALA synthase is the rate limiting enzyme for heme. So ultimately the end product is heme. Okay, now in bone marrow, the heme synthesis needs to be constant because the heme which is produced in the bone marrow is getting formed in the hemoglobin. So hemoglobin production in our bone marrow has to be constant. So that is why ALA synthase 2 has a constant function, whereas ALA synthase 1, which is seen in almost all the cells of the body, mainly in the liver, has variable function. Okay, because the heme here synthesized is one of the utilization is the enzymes like catalase. Okay, so when there is oxidative stress, so these enzymes become more active. For example, the other enzyme is cytochrome P450 enzyme system. Okay, so when we take drugs, these drugs which are metabolized by the liver, so in the liver, this cytochrome P450 enzyme systems are breaking down those drugs. Okay, so when more drugs enter our system, so more amount of these enzymes need to be produced. So heme synthesis automatically increases. Okay, so also the ALA synthase requires coenzymes like vitamin B6 and copper. So what are coenzymes? Coenzymes are the non-protein part of the enzymes which help in the function of enzymes. So if we have a deficiency of B6 or copper, it can lead to anemia. Okay. ALA synthase 1 is also inhibited by the product which is heme. So when the body has sufficient amount of heme, this heme will inhibit the rate limiting enzyme which is ALA synthase 1. Okay. Apart from heme, lead also inhibits ALA synthase 1. ALA synthase 2 on the other hand is inhibited when there is iron deficiency. Okay, because iron is necessary for hemoglobin. If iron is not there, so in the bone marrow, heme production is decreased. Okay, so moving on to next, we have is amino levulinic acid which is produced. So this undergoes dehydration by amino levulinic acid dehydratase. Okay, and water is removed and it leads to formation of porphobilinogen. Now ALA dehydratase is also inhibited by lead. Okay, so lead has important inhibitions on the heme synthesis. Okay, so one place it inhibits is ALA synthase. The second place it inhibits is, the second enzyme is ALA dehydratase, which leads to formation of porphobilinogen. So porphobilinogen is the single pyrrole that we have seen in the previous slide. The single pyrrole is porphobilinogen. Okay, so now we have 4 porphobilinogens. four porphobilinogens combined together, and there occurs a deaminase reaction. Amino group is removed, so we have a deaminase reaction. So the enzyme is porphobilinogen deaminase, and it results in the formation of hydroxymethylbilane. Okay, so this is the hydroxymethylbilane. Then it is a ring structure which undergoes ring closure to form as uroporphyrinogen 3. Okay, so let us understand this. See, here we have four porphobilinogen, 1, 2, 3, 4. Four porphobilinogen, single pyrols. Okay, they are single pyrols. These combine together and they undergo deamination. So when they combine together, they form a ring-like structure which is hydroxymethylbilane. Now this hydroxymethylbilane then undergoes uroporphyrinogen 3 synthase, and it results in the formation of ring closure. Okay, so here we have is ring closure. Okay, and it leads to formation of uroporphyrinogen 3. Okay, so uroporphyrinogen 3 isomer is which is useful in us. Okay, we also have a spontaneous formation of uroporphyrinogen 1 isomer, but that is not significantly useful. So we have is formation of uroporphyrinogen 3 by closure of hydroxymethylbilane. Okay, now this uroporphyrinogen 3 undergoes decarboxylation to form as coproporphyrinogen 3, which again undergoes decarboxylation to form as protoporphyrinogen 9. Okay, so you can remember you have is coproporphyrinogen. So before coproporphyrinogen you have is decarboxylation, and after coproporphyrinogen again you have is decarboxylation. So carbon dioxide is C, coproporphyrinogen is C, so you can remember this way. So once the protoporphyrin 9 is formed, protoporphyrinogen 9 is formed, it undergoes oxidation reaction to form as protoporphyrin. Now protoporphyrin then becomes added with iron. The ferrous form of iron comes and attaches to the ring, and ultimately it leads to formation of heme. Okay, so the enzyme which is required for this addition of iron is ferrochelatase. Okay, and this also is inhibited by lead. So you can see lead has important inhibitions in the enzymes of heme synthesis. So this was about the heme synthesis, how it takes place partly in the mitochondria and partly in the cytoplasm. Okay. Now let us look at the regulation of heme. Now heme is regulated by three important enzymes: ALA synthase 1, ALA synthase 2, ALA dehydratase, and ferrochelatase. Now, as we have discussed earlier, ALA synthase 1, which is seen in liver and most of the other tissues, it is inhibited by heme. So it's a feedback inhibition. Okay, so heme inhibits by repression. Okay, so the genes for the enzyme ALA synthase itself is inhibited. Okay, then hematin, it also has inhibition of ALA synthase 1. Okay, lead, pyridoxine deficiency, and high glucose. So all these inhibit ALA synthase 1. Okay, so high glucose sometimes when the heme production is high, okay, and which leads to toxicity. So we can inhibit the heme synthesis with the help of giving high glucose to the patient. Okay, ALA synthase 1 is induced by drugs. Okay, so these drugs induce reduction of cytochrome P450. Okay, as we have discussed earlier. Now ALA synthase 2, which is present in the bone marrow, it is inhibited when there is iron deficiency. Okay, heme does not have a feedback inhibition for ALA synthase 2. Okay. ALA dehydratase is also inhibited by lead, and ferrochelatase is also inhibited by lead. Okay, so this is how the regulation of heme synthesis takes place. Next we will look at what are porphyrias and what are the various types of porphyrias. Okay, so thank you, and I will see you in the next lecture.